When skeletal muscles are de-innervated, two things happen. The muscle becomes paralyzed, of course, and the acetylcholine receptors on the muscle will spread all over the muscle cell instead of at the end plate where the acetylcholine receptors normally reside. So the muscle-nerve relationship is really strange in a way because they have tropic influence on each other. In this one experiment, they took a fast muscle and they cut the nerve, and then they moved it over to the slow muscle. This causes the slow muscle characteristics to be changed completely, similar to that of the fast muscle from the beginning. Embryonic muscles do not have any nerves, and their acetylcholine receptors are found all over the membrane. Then, during development, when the nerve comes to innervate the muscle, the acetylcholine receptors, besides the one at the end plate, will disappear. So when a muscle is being re-innervated, the same thing happens. All the acetylcholine receptors that have spread throughout the membrane will disappear except for the ones at the end plate. So when the brain sends motor neurons to innervate a muscle, the nerves that do not get to innervate anything shrivel and die, because a muscle that is innervated by one nerve cannot be innervated by another nerve. So when a lower motor neuron dies for some reason, the axon starts to deteriorate, which causes the acetylcholine to start spilling out. The spillage of acetylcholine causes the muscle to twitch, and fasciculation is where there's low frequency twitching, while fibrillation is where there's high frequency twitching. So when you hold something for a very long time, you get muscle fatigue, but before that can happen, you feel pain. This is called ischemic pain, and it's due to the accumulation of byproducts of muscle contraction, like lactic acid. Also, aging muscles lose muscle fibers and decrease in ATP, creatine phosphate, glycogen, elasticity, blood flow, and myoglobin, so therefore they're going to decrease in endurance. Okay, remember that we said that both the skeletal and cardiac muscles are striated, the difference being that the cardiac muscle is going to be involuntary. So the intercalated discs that we see right here of the cardiac muscles, they're going to link the cells of the muscle together, making the cardiac muscle a functional syncytium. And this means that if I were to stimulate one cell, I would either get no response or the entire muscle would contract. There are three types of cardiac muscles: the atrial, which is what makes up the upper part of the heart; the ventricular is the thick lower part of the heart; and then the specialized muscles can either be excitatory or conductive, and they are characterized by weak contraction, rhythmic discharge, and fast conduction. So under a microscope, we can see that the intercalated discs actually separate each cardiac cell. However, they are connected through gap junctions, as we can see right here, and those gap junctions are going to allow molecules and ions to go back and forth. Intercalated discs also have the adherent junctions and desmosomes, in which function to hold the cell together. Without the adherent junctions or the desmosomes, the cardiac cells would actually detach from one another. So every type of cardiac muscle is going to have a different action potential. The atrial and the ventricular are very similar except for the duration of action. So the ventricular is actually going to have a larger duration of action potential. Then there is conductive tissue at the septum between the left and right ventricle, and they have very high resting membrane potentials, as you can see, of -90 to -100 millivolts. Then the pacemaker's action potential is super low, as we can see. It's -55 to -60 millivolts, and this type of low resting membrane potential allows the cell to become rhythmic. So the low membrane potential isn't negative enough to keep the sodium ions from going inside. So it's going to become rhythmic. Right, we already know this, but the 0 right here is going to be due to the rapid entry of sodium ions, and this is depicted by this little red line right here on the second graph. Then number 2, we see a plateau, and this is due to sodium channels closing and calcium ions flowing inside. So during the plateau, notice that the permeability of potassium actually decreases, as we can see from this black line right here. So potassium decreases during the plateau phase, and this is due to the closure of potassium inward rectifiers, or the KIR. And then number 3 right here is just going to be opening of the KDR, or the potassium delayed rectifiers. Right. The P wave is going to be due to atrial contraction. The QRS is going to be due to contraction of the ventricle, and then T right here is going to be due to ventricular relaxation. So the obvious difference between the skeletal and the cardiac muscle is that the sarcoplasmic reticulum, seen right here in the skeletal and then seen right here in the cardiac muscle, is going to be not very well developed, while the transverse tubule within the cardiac muscle is actually going to be better developed, and it's going to be about 5 times wider than that of the skeletal muscle. In addition, the transverse tubule of the cardiac muscle contains a polysaccharide that has the ability to store calcium, and there are more mitochondria in the cardiac muscle. Contraction of the cardiac muscle and an action potential is generated in the cardiac muscle. And just a side note, the cardiac muscle does not need a nerve to generate that impulse. So the action potential is going to be generated in the cardiac muscle, and it depolarizes the membrane, including the transverse tubule. Then calcium channels are going to open and calcium goes in. Then the calcium is going to activate the DHPR, which then activates the ryanodine receptors to release more calcium for storage. So in skeletal muscles, calcium comes largely from the outside, while in the cardiac muscles, calcium comes from the outside and the inside. So the contraction of cardiac muscle depends on the concentration of calcium on the extracellular fluid. So how do we remove calcium in order to end contraction? The calcium pumps calcium outside into the mitochondria, and then into the transverse tubule, and then the cycle is going to repeat. So because of the structure of the cardiac muscle, contraction is almost like the all or nothing law. Basically, if you have enough stimulus, then you have the entire muscle contract. So you cannot tetanize a cardiac muscle because it has a very long refractory period and a super short relative refractory period. So this is the muscle fiber length and tension relationship, and this graph is basically telling us that the more you stretch a muscle fiber, the more force of contraction you get, until you reach a point where the force of contraction is actually going to decline, as we can see right here. So this is going to be seen in heart failure, where the ventricles fill with blood and then stretch, and then at a certain point they lose their force of contraction. The inotropic effect is the force of contraction. Catecholamines, like epinephrine, have a positive inotropic effect, which means they increase the heart's force of contraction. And this works using cyclic AMP, which supplies more calcium to the heart. So most of the energy that is used for cardiac contraction comes from the fatty acids that we eat. And during starvation, adipose tissue is broken down to supply the heart with energy. The sinus node, seen right here, or the SA node, is going to attach to a bundle of similar cells that go to the AV node. And from the AV node, we have a bundle that goes into the septum between the left and right ventricle called the bundle of Hist. And then at the apex of the heart, the bundle of Hist will branch out into the outer walls like this, and this is making the Purkinje cells. So chronotropic refers to the rate of contraction and is driven by the SA node, where all of the action starts. And then dromotrophic refers to the speed of conduction of the action potential. So just remember that the pre-potential, seen right here, is going to be caused by many mechanisms like tiny calcium channel transient, or the opening of potassium channels, which makes potassium leave, and then the inside of the cell becomes less negative, causing the threshold to be reached. And once that happens, we see an upstroke, and this is due to the influx of calcium ions. So like we said, the heart has a driver, and that is the SA node. The cholinergic nerve comes from the vagus, which supplies the SA node and the AV node, and then the adrenergic innervation supplies mostly the ventricular tissue. The function of these nerves is to modulate the heart, so if we injected adrenaline into the heart, the force of contraction will increase. And then if we stimulate the vagus nerve, it will release acetylcholine on the SA node, which will act on the acetylcholine receptors, which will cause slowing of the heart. So in other words, if you stimulate the vagus, you will have a negative chronotropic effect, meaning that the rate of contraction will decrease. Whereas if you block the adrenergic receptors of the heart, you will have a negative inotropic effect, meaning that the force of contraction will decrease. So the smooth muscles are very small in size and contain no striations and are very poorly developed sarcotubular systems. There are two types of smooth muscles. The unitary are functional syncytial, and the multi-unit are independent of each other. In the multi-unit, notice that each cell has its own nerve supply. The unitary have things called the nexus, which are similar to intercalated discs because they have a lot of gap junctions that permit coordinated contraction. And then the multi-unit muscles are heavily innervated and have no spontaneous activity. So just remember that the resting memory potential for smooth muscles is very low. It's going to be negative 55 to negative 60 millivolts. The majority of the action potentials are seen in spikes, as seen right here. But we also see the plateau type in the uterus, and then slow waves do not produce an action potential until they reach the threshold. So for the smooth muscle action potential, depolarization or the upstroke is due to calcium entering, repolarization is due to potassium exiting, and remember that sodium is not as abundant as calcium in the smooth muscles, so it's not going to be involved in the action potential, but sodium can facilitate the opening of calcium channels in the slow wave. So interstitial cells of Kajal act as drivers of the slow wave potential, and automatic nerves have beads of varicosities that contain neurotransmitters and modulate the activity of the slow wave. So smooth muscles do not have striations or sarcomeres as we know it, but they do contain actin with tropomyosin, but no troponin. And there is more actin than myosin. The dense bodies are where the actin and myosin come out. As you can see right here, the dense body is that really dark spot. That's where the actin and myosin come out. And then the caveoli are like transverse tubules, except they're not as deep as we can see right here, because the smooth muscle is too small, so it doesn't need that extra length. So the smooth muscle gets its calcium completely from the extracellular fluid. When calcium enters, it is able to diffuse to all parts of the cell because the smooth muscles are so small. However, the time that it takes the calcium to diffuse is really slow. It's going to be about 200 to 300 milliseconds before we can see contraction. And this is called the latent period. Calcium is removed by calcium pumps in order to stop contraction. However, that takes a really long time too, which is why the duration of smooth muscle contraction is so long. So instead of troponin, we see calmodulin in the smooth muscles. Calmodulin is activated by calcium, which interacts with myosin in the light chain kinase. The kinase will phosphorylate the myosin head and cause it to contract. So the multi-unit smooth muscles can contract without an action potential. All they really need is a small depolarization to start contraction mechanism, which basically means supplying a little bit of calcium. And then denervation of a smooth muscle causes it to become hypersensitive. However, they do not atrophy like skeletal muscles do. The loss of norepinephrine uptake mechanism causes hypersensitivity.